Hi, everyone. My name is Floyd Nichols, and I'm currently a fourth-year PhD candidate working with Maggie Osborne. And today I'll talk a bit about some of my primary work, which is looking at lipid biomarkers in Martian analog lakes. So to set the setting for my study sites, I study these hypersaline lakes in the interior of British Columbia. These hypersaline lakes are very unique in that they have extremely high sulfate concentration, shown here. So on the y-axis, I've got log sulfate concentrations, and on the x-axis, just the settings of different aqueous systems. So you can see in this point circle here, which are the lakes that I study, they have orders of magnitude higher sulfate concentrations than the sea, for instance, and even other high sulfate systems. And so this is great for probing questions when thinking of paleo lakes on Mars that were probably extremely sulfate rich, as well as moons, icy moons like Enceladus or Europa. And so throughout the last year or so, I use these lipid biomarkers to get an understanding of how life might be preserved and or the production potential as well for some of these biomarkers in these systems. And we see actually that in these red rectangles, that the primary lipids that we see are actually microbial dominated, as opposed to other things that could be terrestrially derived, for instance. And so this is very different than what we've seen in other astrobiological analogs systems. And so it's like super cool. However, one of the limitations with my work currently is we use traditional methods of gas chromatography mass spectrometry. And that only allows us to view a small portion of the structure of the lipid biomarker. And so if we can see the entire structure of the lipid biomarker, we can actually get more information, something that might be more closely related to, say, DNA analysis. And so part of my work this summer has been learning how to use a new instrument in the MSERC department here at Northwestern, which is called an HPLC QTOF MS, which stands for High Performance Liquid Chromatography Quantitative Time of Flight Mass Spectrometry. And so this technique allows us to view the entire structure of the lipid biomarkers in our systems. And so we can see here each individual point relates to a individual compound. And just from this instrumentation here, we can already see a vast majority of compounds in our samples, more so than we could have seen using traditional methods with GCMS. And so the other great thing with this data is because we can actually see the entire structure of the lipid biomarker, we can do a similar analysis that Matt actually mentioned earlier, which is using a combination of information theory and molecular networking to actually get an idea of how these sort of perhaps unknown compounds relate to each other. And so, as Matt mentioned earlier, a single individual node represents an individual lipid, although in that case, it was a microorganism, and a cluster represents lipid classes. And so this is great for this type of work because in the previous slide, I showed so many different points, and it's going to be extremely difficult to actually analyze manually, which is what we do with GCMS. Additionally, if there are points such as these nodes that are not necessarily clustered, they might represent novel lipids, so this instrument or this technique will also allow us to potentially discover new lipids that are in environmental settings. And that's all.
I'm curious, great talk, Floyd, but I'm curious if you have had a chance to like compare this new method to some of the CMS like analysis that you've done. Like, have you done that on the same sets of samples?
No, that's a great question. So this is super preliminary work, as in I just got this data like last week. And so we're still in the process of streamlining this process. So we only have a couple samples right now. We haven't run all of the samples. So hopefully in the next couple of weeks, I'll have a better idea of how that compares to GCMS.
I guess on that, are you able to do that here? Like,
Yeah, so I'm doing it in tech. Down at there's a core facility in the G wing.
Okay, cool.
Thanks, Floyd. this is gonna be some kind of proxy for ancient Mars, then do you see that as a possibility that like those biomarkers could go to live on Mars and bring them back?
Yeah, that's a great question. So I think there's a utility to using both an HPLC MS as well as GCMS. So GCMS is the more traditional method. HPLC is what gives us this more detailed look of the lipid biomarker. So when looking at using the traditional method, we're actually looking at more stable molecules, which are more likely to be preserved on geologic time. Whereas with the HPLC, because we're actually looking at the full lipid structure, which is also going to contain some more labile components in the lipid biomarker, they might not last as long on geologic timescales. But that's part of this work too, is we also have sediment cores. So we want to see how far down in geologic time these things actually are preserved.